What you just observed was a very artistic articulation of coral bleaching, but there was a lot going on there, so let's rewind and break down our characters. Once we understand coral bleaching a bit more, I would love to share with you some of the research I have been doing on it. So first, we have our coral, which make up coral reefs. Next, we have our dinoflagellate, which is a photosynthetic protist, a tiny organism which can harness energy from the sun. These two organisms live in a symbiotic relationship, and the dinoflagellate actually lives inside the coral cell. This symbiotic relationship is a mutualism, which means that it is beneficial for both parties. For example, the dinoflagellate, which I mentioned before is photosynthetic, can harness energy from the sun and then give that energy to the coral, and the coral is able to take the energy and provide a safe home for the dinoflagellate along with other nutrients. Now to the ominous dancers in the back. These are our stressors that are currently threatening coral reefs. First, we have ocean acidification. This is caused by the rising levels of carbon dioxide that is absorbed by the ocean. Next, we have pollution. Pollutants are washed away by rain and carried down through streams and rivers to the ocean. Pollutants which can harm corals include sediment from deforestation, agriculture, and development, toxins from sunscreen and oil spills, pathogens from spilled sewage systems, and increased nutrients from fertilizer. And finally, we have an increase in ocean temperature. To explain this, we gotta talk about El Niños. The ocean goes through seasons, just like Georgia does, and there's a time where the part of the ocean is warmer. These events are called El Niños. Due to climate change, El Niños have become increasingly hotter each season, and these intensified El Niño events are the main driver of coral bleaching. All of these stressors disrupt the balance between the two partners in this symbiosis, causing a phenomenon called coral bleaching, in which the dinoflagellate separates from the coral cell. The coral, now lacking its main food source and still experiencing stress, usually perishes. Now you might be asking, SK, why does this matter? And I would tell you that corals are very important. Here's why. If you remember at the very beginning, I mentioned that corals make up coral reefs, and coral reefs are very important to humans. So let's take a look at a coral reef and its benefits. First, coral reefs provide protection against storms like tsunamis. They also provide food sources to communities who live on the coast and those populations which eat fish. A third reason why they're important is because they provide a haven for ecological diversity, which provides stability to the ecosystem. And lastly, coral reefs are very beautiful. But all these benefits can be lost during a bleaching event, because not only one coral can die, but an entire coral reef ecosystem can die. This causes the once ecologically diverse ecosystem to disappear. I just explained to you the basic process of coral bleaching. But there's so much more going on behind the scenes on a molecular level, a lot of which scientists have yet to discover. And this is where I come in. Through my time at Berry, I have been researching what bleaching looks like on a cellular level. Specifically, I am interested in the protein interactions which are involved in this process. To do this, I used a model organism called Aptasia, which is an anemone, and it engages in the same symbiotic relationship with dinoflagellates that corals do. Yet Aptasia are much easier to take care of in a lab setting and are easier to manipulate for experiments. The first step to testing protein interactions is to extract the Aptasia's RNA. But to understand why you need to do this, you first must understand the central dogma of biology. The central dogma is a basic scientific process which is fundamental for all of life. And it starts here, inside the nucleus of a cell with your DNA, the blueprint for cellular function. An enzyme called RNA polymerase then comes and reads the blueprint of your DNA and rewrites it as RNA, which can be transported out of the nucleus. The RNA copy can then be used by ribosomes to make proteins, which carry out a variety of different functions in the cell, such as providing structure, fighting diseases, and even turning on other genes to start the process over again. Thus, now we understand DNA leads to RNA, and RNA leads to proteins. Good. Back to our anemone, aptasia. I said I extracted the RNA from it, but there's a problem with RNA. It's super unstable. So we can actually turn it into DNA, a much more stable molecule using a fun enzyme called reverse transcriptase. Now we have a bunch of DNA from our aptasia. But I wanted to test specific protein interactions that we believe might be involved in coral bleaching. Specifically, there are two groups of proteins we are looking at: GMAPs and BCL2 proteins. Not much is known about GMAPs in invertebrates, organisms without spines, like our aptasia and corals. But we know that they are responsible for immune responses in vertebrates, organisms with spines like humans. BCL2 proteins, on the other hand, are proteins responsible for apoptosis, a fancy term for programmed cell death. This process, apoptosis, is an important part of coral bleaching. Therefore, we must understand what goes on during apoptosis to understand coral bleaching. Scientists do know that GMAPs and BCL2 proteins interact inside of vertebrates, things with spines. So I wanted to test if aptasia GMAPs and BCL2 proteins interact to help us better understand the molecular process of coral bleaching. So how am I going to test if two proteins interact with a bunch of DNA that I now have? If we remember that DNA codes for RNA and RNA for protein, we can start to see a way forward. The proteins I am interested in have specific DNA sequences, which we know from past research, and these sequences are somewhere in the DNA sample that we have extracted. So, by cool science called PCR, I'm able to amplify just the select sequences that I am interested in and isolate them from all other DNA. Now, in order to get from this DNA sequence to testing protein interactions, scientists can use yeast. Yes, this fungus is an unlikely solution, but let me explain this method called the yeast-2 hybrid. In this protocol, we use two different strains of yeast, and we take our genes of interest that we have isolated and put them into the two different yeast strains. Next, we grow these two yeasts together and they will join, creating a new hybrid yeast strain with both of our genes of interest inside. So, at this moment, we have genes from our aptasia that are currently inside of a yeast cell. And if we recall from the central dogma, that DNA turns into RNA, which then turns into proteins, and in the yeast cell, the aptasia DNA codes for the proteins we want to test. So, really inside of our mated yeast, we have our two proteins, which we wanted to test the interaction between. The next step is to take our mated yeast and grow it on special media, so that if our two proteins interact, the yeast will turn blue. But if the proteins don't interact, the yeast will not survive. So in summary, blue yeast, the proteins have interacted. No yeast, the proteins did not interact. Now, you might be wondering, did it work? Did GMAPs and BCL2 proteins interact like we thought they would? Well, our results were in fact not what we expected. I started by testing GMAP and BCL2 interactions, and even GMAP-GMAP interactions, but with no luck. All the yeast died, and so no protein interactions were proven. But then, last December, I got one of the best Christmas gifts.
Merry Christmas!
Blue yeast! But I wasn't even testing GMAP-BCL2 interactions. I had already had lots of the genes in the yeast strains, so I actually decided to also test BCL2-BCL2 interactions. This time, I was using a new BCL2 gene called BAC, and the protein it codes for interacted with other BCL2 proteins. In fact, it interacted with MCL1, BCLWA, and BCLWB, which are all BCL2 proteins. These results correlate to past scientific research in Hydra, an organism which is similar to Aptasia, and corals. It has been shown that BCLWA and BAC interact like we found. But we were actually the first to show that MCL1 and BAC interacted, as well as BCLWB interacting with BAC. This shows that we confirmed interactions shown in past scientific research, but we also found novel interactions never found before. To make sense of all of this in Aptasia, let's look more closely at the functions of these BCL2 proteins. In most animals, BAC has been characterized as a protein which causes apoptosis, while MCL1, BCLWA, and BCLWB are proteins which stop apoptosis. So we think that MCL1, BCLWA, and BCLWB proteins are all produced to bind to BAC and prevent it from causing apoptosis. But when MCL1, BCLWA, and BCLWB are not produced, BAC is able to function and cause apoptosis. Look, we learned more about how apoptosis works, which in turn tells us a little bit more about coral bleaching. But what about GMAPs? It looks like they might not play a part in coral bleaching. But even though the yeast-2 hybrid showed no evidence of interaction, the relationship with these proteins is anything but solved. Here are just a couple scenarios in which GMAPs might interact with BCL2 proteins, but it couldn't be proven using the yeast-2 hybrid. One possibility could be that there's an intermediate protein that facilitates the interaction between these two proteins, which wasn't present because we had isolated them and put them into the yeast. Another possibility is that this potential intermediate protein could have been another GMAP protein. We know that sometimes GMAPs like to form dimers, which means that they interact with themselves, and a BCL2 protein could have interacted with this dimer. But why are we so set on finding the relationship between these two proteins? It's because past research has shown that GMAPs are somehow correlated with apoptosis. And as we know from our high school statistics class, correlation does not equal causation. You have to have more data to show a causal relationship, and that is what we are trying to provide. And another variable, just to make things a bit more confusing, is that GMAPs in Aptasia are all what we call short GMAPs. But in corals, there are short GMAPs, but there are also long GMAPs. And these long GMAPs might interact with BCL2 proteins. But we were only working with short GMAPs from Aptasia. So where does this leave us? We did find out a little bit more about BCL2 proteins in Aptasia, but we're still at a loss for GMAPs. But don't worry. More research projects are being worked on right now to help us learn more about the mystery role of GMAPs. Maybe I can help those scientists use the answers to explain their research too. Now we come to the part of the show where you get to ask me questions. and I will get to answer them while choreographing on the spot with my dancers. It should be a fun time, and you'll get to see a little bit into the process of SK's rehearsals. So, I'm ready for questions.